Once upon a time, in a kingdom not so far away, lived a king and queen. The king was short and stout, while the queen was tall and slender. One day they had a baby, and much to everyone's surprise, the baby was short like the king. Everyone was confused because the baby looked nothing like the queen. So, how did he inherit the king's short stature? Well, the answer lies in the science of heredity. In this Science Show special, we'll take you on a magical journey through the world of genetics, exploring the fascinating history of this captivating field. Join us as we uncover the secrets of genetics and discover the incredible discoveries that have shaped our understanding of heredity. Thousands of years ago, people observed that offspring resemble their parents, but they didn't understand why. They just knew that if they picked plants or animals with desirable traits like bigger fruits or faster speed, those traits would be passed on to the next generation. This led to the practice of selective breeding, where people would choose which plants or animals could reproduce based on their desired traits. For example, farmers would breed cows that gave the most milk or grow plants that produced the biggest yields. However, people didn't know why certain traits were passed down until the 1800s, when a monk named Gregor Mendel conducted some experiments. Mendel experimented with pea plants by crossbreeding plants with different traits such as color, shape, and size. He carefully analyzed the results and discovered that traits are transmitted by units called factors, now known as genes. He also uncovered two key laws of inheritance, the law of segregation and the law of independent assortment. The law of segregation states that each individual carries two copies of a gene and these copies separate during gamete formation, with only one copy of each gene passing on to the offspring. The law of independent assortment states that genes for different traits assort independently of each other during gamete formation. This means that the inheritance of one trait does not affect the inheritance of another trait. Mendel's work laid the foundation for the field of genetics. However, his findings were not appreciated until around 1900 when three scientists, Hugo de Vries, Karl Correns, and Erich von Tschermakry, discovered his work. They recognized the significance of Mendel's laws and built upon his findings, leading to the establishment of genetics as a separate branch of biology. After Mendel's work was rediscovered, scientists began to understand that genes are located on structures called chromosomes. This led to the development of the chromosome theory of inheritance, which states that genes are carried on chromosomes, and it is the behavior of chromosomes during cell division that explains the patterns of inheritance observed by Mendel. Scientists like Walter Sutton and Theodore Boveri provided evidence for this theory by studying the behavior of chromosomes during meiosis. Thomas Hunt Morgan further established this theory by experimenting on fruit FLIs and showing that genes are linked together on the same chromosome. He also discovered that genes can be transferred between chromosomes through a process called crossing over. As the understanding of genetics grew, scientists began to unravel the chemical composition of genes. In 1944, Oswald Avery, Colin MacLeod, and Macklin McCarty demonstrated that DNA is the genetic material. They conducted experiments showing that DNA from a non-virulent strain of bacteria could transform a virulent strain, indicating that DNA is the molecule responsible for heredity. In 1953, James Watson and Francis Crick, building on the work of other scientists, including Rosalind Franklin and Maurice Wilkins, revealed the double helix structure of DNA. This discovery provided an explanation for how genetic information is stored and replicated, and it revolutionized our understanding of genetics. With the discovery of DNA structure, scientists were able to determine the chemical basis of genes. In the 1960s, Marshall Nirenberg and Heinrich Mattei cracked the genetic code, deciphering how DNA sequences translate into amino acids during protein synthesis. This discovery allowed researchers to understand how genetic information flows from DNA to RNA to protein. The 20th century saw remarkable progress in genetics research. In 1973, Paul Berg combined DNA from different sources, creating the first recombinant DNA molecule. This technique laid the foundation for genetic engineering and biotechnology. As our knowledge of genetics expanded, scientists developed techniques to manipulate DNA, leading to the creation of genetically modified organisms. One of the most significant achievements in genetics was the completion of the Human Genome Project in 2003, which successfully mapped the entire sequence of human DNA. This project provided a wealth of information about the genes and genetic makeup of humans. In recent years, advances in DNA sequencing and biotechnology have led to the development of gene editing tools such as CRISPR. CRISPR technology allows scientists to precisely modify DNA sequences, opening up new possibilities for gene therapy and genetic engineering. Today, genetics plays a crucial role in various fields, including medicine, agriculture, and biotechnology. It has helped us understand the genetic basis of diseases, develop new therapies, and enhance crops through genetic modification. As our knowledge of genetics continues to grow, it holds immense potential for shaping the future of biology and medicine. The field of genetics has come a long way since Mendel's initial experiments with pea plants, from the discovery of DNA as the genetic material to the development of gene editing tools. Each milestone has brought us closer to understanding the complexities of heredity. As we continue to explore the mysteries of genetics, we can appreciate the incredible discoveries that have shaped this fascinating field.